Today we'll be starting with lipid chemistry. We'll be discussing fatty acids and triacylglycerols. Lipids are lipid word comes from lipos meaning fat or grease. They are substances of biological origin which are soluble in organic solvents but insoluble in water or sparingly soluble in water. Fats, oils, certain vitamins and hormones, most non-protein membrane components are lipids. Body lipids are mostly intracellular or part of the cell membrane. They are transported in blood as water-soluble lipoproteins. So there are five major classes of lipids. MBBS students of Maharashtra University, they are asked to write classification of lipids in the exam. At the end of this lecture series, I will tell you how to write classification or what to write in classification of lipids in the exam. But we will be in these lectures understanding these five major classes of lipids conceptually. Starting with fatty acids. Fatty acids are carboxylic acids with long chain hydrocarbon side chain. So there is a carboxyl group and a hydrophobic hydrocarbon chain. They are mostly esterified, rarely free, and mostly they are C16, C18. And most of the fatty acids in the body have even number of carbon atoms because they are formed by concatenation of C2 unit. Concatenation is a process of joining sequential objects end to end to form a single continuous series. Fatty acid consists of a hydrophobic hydrocarbon chain with a terminal carboxyl group that has a pKa of 4.8. Now, what is the pKa of fatty acid? pKa of fatty acid is the pH at which 50% of the molecules are neutral and 50% of the molecules are in deprotonated or negatively charged form. It is an estimate of how easily the fatty acid will lose its proton. Lower pKa means a stronger acid, greater tendency to lose proton. Short and medium chain fatty acids are absorbed directly through the gastric mucosa in the bloodstream because in the acidic environment of the gastric acidic environment, they are neutral and so they diffuse through the cell membrane. Increasing the number of double bonds in a fatty acid decreases its pKa, making it a stronger acid, and increasing the chain length will increase pKa, making it a weaker acid. So physiological pH is 7.4, and at that pH the terminal carboxyl group ionizes and the ionized form is the predominant form. And the anionic group is what makes it uh, amphipathic. That is, it is both hydrophilic and hydrophobic. Fatty acids are both hydrophilic and hydrophobic. However, the longer is the chain, carbon chain, they become more and more hydrophobic because the hydrophilic part is only that deprotonated carboxyl end, whereas the alkyl chain is hydrophobic. Longer chain fatty acids are hydrophobic portion is predominant. They are water insoluble and they should be fatty acids must be transported within with a protein. Most of the fatty acids in plasma are in fatty acid esters in lipoproteins. About 10% fatty acids are free and they are circulated with albumin. Fatty acids can be short chain, medium chain, long chain, very long chain. Each of these have their physiological functions. Short chain, they are formed by gut bacteria through fermentation of dietary fiber. They can be acetate, propionate, butyrate, and about 60 to 75% of the energy requirement of the colonic cells comes from these short chain fatty acids, mainly butyrate. However, small intestinal cells utilize mainly glucose for their energy. Short chain fatty acids also help in regulating inflammation and aid in metabolic function. Medium chain fatty acids. Short and medium chain, we saw that they are absorbed directly through the stomach mucosa. They are not packed into lipoproteins, right? And these medium chain fatty acids are metabolized rapidly for energy in the liver. Long chain fatty acids are foundational for human life because they play a very important role. They are part of cell membrane. They are vital substrates for energy production via beta oxidation in the muscle, heart, and liver. We all know that fatty acids are predominant energy source of the heart. That is, heart utilizes mainly fatty acids for its function, myocardium. And so, just by living, even as you are resting, even in the resting state, heart utilizes fatty acids mainly. And long chain fatty acids are crucial precursors for hormone like signaling molecules. So icosanoids are hormone like signaling molecules. They are local hormones, autocoids. They are paracrine signaling. I mean, they are formed from 20 carbon fatty acids like icosapentaenoic acid and arachidonic acid. We'll see in the next slide. And so these icosanoids, they are paracrine signaling. Then endocannabinoids like anandamide. You can read about this anandamide. It's a natural antidepressant molecule, and you can actually stimulate the release of these endocannabinoids, anandamide, by exercise and pranayama and stuff, and you can come out of depression. Docosanoids like resolvins, protectins are anti-inflammatory and neuroprotective. Long chain fatty acids can be essential fatty acids. They must be obtained through diet. Body cannot synthesize them. Essential fatty acids are alpha linolenic acid and linoleic acid. And these docosahexaenoic acid, icosapentaenoic acid, arachidonic acid, which are structurally functionally important, they can be synthesized from the essential fatty acids, right? And the docosapentaenoic acid. Now, linoleic acid, linoleic are omega 3, omega 6. Now, which of the two is omega 3 and omega 6? That you can read up, that's your homework, and you can even post it in the comments. Structurally, functionally important long chain fatty acids, docosahexaenoic acid, right? It plays a role in cognitive function, visual health. Icosapentaenoic acid is anti-inflammatory. I mean, it is used in synthesis of omega-3 icosanoids, which are anti-inflammatory. It's found mainly in fish oil. Arachidonic acid is found in cell membrane and phospholipids, and it's a direct precursor to inflammatory mediators. You will learn so much about this even in pharmacology next year. So icosanoids are locally acting signaling molecules derived from 20 carbon polyunsaturated fatty acids like arachidonic acid, icosapentaenoic acid. Functioning as local hormones, they act on nearby cells. They are crucial for regulating inflammation, immune responses, fever, blood pressure, blood clotting. Very long chain fatty acids are structural lipids with carbon chain of 22 carbon or more. They are crucial for skin barrier formation, liver homeostasis, brain function. They require strict regulation in the sense that, see, they are synthesized in the endoplasmic reticulum and they are degraded in the peroxisomes. But in peroxisome biogenesis disorders, which are genetic disorders of peroxisome function, the peroxisome is not able to degrade them, and so that leads to accumulation of very long chain fatty acids in the peroxisome, and that leads to signs and symptoms of these disorders like X-linked adrenoleukodystrophy or Zellweger spectrum of disorders. Very long chain fatty acids cannot be oxidized by the mitochondria. They need to be initially oxidized in the peroxisomes. We have discussed this at length in the video on the cell. And Physiological roles of very long chain fatty acids are that they play a role in membrane architecture by being part of sphingolipids and glycerophospholipids. We'll be discussing those in the next video. And ultra long chain fatty acids like hexacosanoic acid, montanic acid, tetracosanoic acid are vital for specialized tissues including retina, testes, meibomian glands. And lignoceric acid, hexacosanoic acid, they are part of myelin sheath and hence useful for nervous system health. And very long chain fatty acids also are part of skin ceramides that prevent water loss. So these are some common biological fatty acids. You see that the number of carbon and the number of double bonds. So this 12 colon 12 colon 0, it means that there are 12 carbon atoms here in this molecule and no double bonds. 16 colon 1 means that there are 16 carbons and one double bond. And N7 means that the double bond is from the seventh carbon from the terminal carbon atom. Terminal carbon atom is the methyl terminal. Methyl terminal is also known as the omega end of the chain. And seventh carbon from the methyl terminal is the double bond. 18,3 N3. So N3, N and omega can be considered as equivalent. We'll see later on, right? N3 means that the double bond is third carbon from the terminal carbon, and there are two other double bonds. We'll see that later in the next nomenclature. So convention is that chain length and number of double bonds are separated by colon, like 16 is to 0. If there is one double bond, it's shown like this, right? This is the structure of oleic acid, which is an 18 carbon fatty acid with a single double bond between 9th and 10th carbon atom, and that is written as 18 colon 1 and delta raised to 9. So delta raised to 9 means that double bond is between the 9th and 10th carbon. The number here suggests the lower carbon from the carboxyl end. And in case of polyunsaturated fatty acids, we can start numbering even from the methyl terminal. The methyl terminal carbon is known as omega carbon, and we start numbering from there. And right, besides the physiological role, PUFAs uh, they are related to position of first double bond near. Yes. Very importantly, why do we start numbering the PUFAs from the omega terminal end? Is because the position of the first double bond near methyl end of the chain, right? It kind of has something to do with the physiological role of the polyunsaturated fatty acids, right? Than the double bond that is near the carboxyl end. And that is why the alternative nomenclature is used for these fatty acids. Carbon of the methyl group, the carbon most distant from the carboxyl group is omega. Omega is the last letter in Greek alphabet. There are 24 letters in Greek alphabet, and surprisingly zeta is just the sixth letter. Carbon is given, this methyl terminal carbon is called omega carbon. It is given number 1 in polyunsaturated fatty acids, where their physiological role is related to position of the first double bond near the methyl end of the chain. Position of double bonds are indicated relative to omega carbon. In this convention, PUFAs with double bond between C3, C4 are called omega 3 and between 6 and 7 are omega 6. This is icosapentaenoic acid, right? Designated as 25 delta 5, 8, 11, 14, 17. So there is a double bond between 5th and 6th, 8th and 9th, 11, 12, 14, 15, 17, 18. Right? And 5 double bonds, obviously. And is also an omega 3 fatty acid, emphasizing the biologically important double bond in the omega 3 position. Right? Arachidonic acid. Again, this is showing arachidonic acid. Right? Omega 6 fatty acid, because first double bond from omega is 6 carbons from that end, also referred to as N6, right? We saw that omega and N are equivalent. Now, something to note here is that triple bonds are rarely seen in any compound of biological origin, and fully extended conformation of saturated fatty acids is highly stable, and melting points of saturated fatty acids increase with molecular mass. If we see in this table, saturated fatty acids melting point is increasing as the molecular mass is increasing. Fatty acids become fatty acid double bonds almost always have cis configuration. That is, the two groups are on the same side of the double bond. If they are on the opposite side, it is called as trans configuration. And this cis configuration puts a rigid 30-degree bend in the hydrocarbon chain, the unsaturated fatty acid, and interferes with their efficient packing to fill space. We'll see that in the next diagram. Consequently, reduced van der Waal interaction cause fatty acid melting points to decrease with the degree of unsaturation. So more the number of double bonds, lesser will be the melting point. We see here in this table again that this palmitic acid melting point is minus zero point 0.5. So even at 0 degrees Celsius, palmitolic acid rather, palmitolic acid even at 0 degrees Celsius is liquid, right? But oleic acid, because number of carbons increased, the melting point has increased. But linoleic acid, add one more double bond, and suddenly, despite having the same number of carbon atoms, 18, right? Oleic acid has a melting point of 13.4, and linoleic acid has a melting point of minus 9. And linolenic acid, add one more double bond, and despite having the same number of carbon atoms, has a melting point of minus 17. And you can see these arachidonic acid has a melting point of minus 49. Icosapentaenoic acid has a melting point of minus 54. And nervonic acid, you can see. So if oleic acid 18 carbon 1 double bond has a melting point of 13.4, nervonic acid at 24 carbons has a melting point of 39. So around room temperature, around atmospheric temperature is 32. Nervonic acid will be solid. Nervonic acid is found in the brain. Mm. So lipid fluidity increases with degree of unsaturation of the component fatty acids. This phenomenon has important consequences for membrane properties. You see here, single bond molecules can rotate freely about the single bond. But if there is a double bond, even one double bond, and that too cis configuration, so it introduces a kink in the structure and prevents the molecules from packing tightly, and that is why it has a lower melting point. This is again showing the same thing, right? This also is showing the same thing. Yes. And if the fatty acid has two or more double bonds, they are always spaced at 3 carbon intervals, right? You see here there is one double bond, then first, second, third carbon, next double bond. Then first, second, third carbon, next double bond. We see in this structure here, icosapentaenoic acid, right? Double bond is at the third carbon. This is the first carbon, second carbon, third carbon, double bond. First carbon, second carbon, third carbon, double bond. First carbon, second carbon, third carbon, double bond. So double bond is after every 3 carbons. And because membrane lipids typically contain long chain fatty acids, the presence of double bonds in some fatty acids helps maintain the fluid nature of the lipids. Fluid nature of the membrane is helped by the double bond present in these fatty acids. Coming to triacylglycerol, they are triesters of glycerol, as you can see in this diagram. They function as energy reservoirs in animals. And They are therefore the most abundant class of lipids, even though they are not components of biological membranes. Most abundant class of lipids in humans is triacylglycerol. Triacylglycerol differ according to identity and placement of the three fatty acids. They can be simple if all three are the same, and they can be mixed if there is more than one type of fatty acid. And most of the fatty acids in our body, most of the triacylglycerols in our body are of the mixed type. We can see here the structure of steroyl, linoleoyl, palmitoyl, glycerol. Yeah. And so if it's mixed, then that is how you write the name. That is, you starting from the first carbon, steroyl, linoleoyl, palmitoyl, glycerol, you write the name. Now, how do you decide which is first carbon, which is second carbon, which is third carbon? Why can't it be 1 palmitoyl, 2 linoleoyl, 3 steroyl? Will it make a difference? Well, you can read about it on Google, but it has something to do with enzyme specificity. At the time of fatty acid synthesis, this enzyme, glycerol 3-phosphate acyl transferase, it prefers saturated fatty acids. I mean, the first atom of glycerol to be esterified with a fatty acid, that is called as SN1 or C1. SN1 position is the first carbon atom esterified during triacylglycerol. First carbon atom esterified during triacylglycerol synthesis is SN1 by glycerol 3-phosphate acyl transferase. And that enzyme generally prefers a saturated fatty acid, right? And for C1, for SN1 position. And that is why in this compound, stearic acid is saturated. So this will be 1 stearoyl, 2 linoleoyl, 3 palmitoyl. And so that nomenclature depends on which fatty acid was added first, right? That is the SN1 position. Second added was SN2 and third was SN3. And the composition of triacylglycerol in the body, like the body, whatever triacylglycerols are made in the body, right? That will depend on the diet, the fatty acids that have come from diet, what kind of fatty acids are there in the cell at that moment. That will determine the composition of triglycerides synthesized by that cell. And now triacylglycerols are hydrophobic because both the polar hydroxyl group of glycerol and polar carboxylate to the fatty acids bound in ester linkage. And lipids have a lower specific gravity than water. We know that oil floats on water. Fats and oils, what are fats and oils? Fats are solids at room temperature and oils are lipids which are liquid at room temperature, right? Plant oils are richer in unsaturated fatty acids than animal fats and they have a lower melting point than animal fats. Now, what is the advantage of having triacylglycerol as a storage form of fat over glycogen as storage form of fat? Because fats are less oxidized than carbohydrates, right? They literally have much less number of oxygen than carbohydrates, and that is why less oxidized means they have higher potential to get oxidized, and that is why they will yield more amount of energy on oxidation. And fats also being non-polar, they are stored in anhydrous form, whereas glycogen binds twice its weight of water under physiological conditions, and therefore fats provide about 6 times the metabolic energy of an equal weight of hydrated glycogen. Right. And in animal cells, adipocytes are specialized for synthesis and storage of triacylglycerols, whereas other types of cells have only a small droplet of fat in their cytosol. So even other cells can have some fat in their cytosol, but adipocytes are specialized for storage of fat, triacylglycerol, in the body. Adipocytes may be almost entirely filled with fat globules. We'll see in the next the photo of adipose tissue. Adipose tissue is most abundant in subcutaneous layer and in the abdominal cavity. Fat content of normal humans about 21% body fat is normal. mm, About 21% body fat is normal for men and 26% for women. It enables us to survive starvation for about 2 to 3 months, whereas glycogen stores of the liver get depleted in about 24 hours. Subcutaneous fat also provides thermal insulation, which is important for warm blooded animals like humans and also animals like whales, seals, geese, etc., who are routinely exposed to lower temperatures. This is the same thing from a different resource, and you can see that despite triacylglycerol being a storage form of energy in the humans, carbohydrates are the main fuel because they are a quick source of metabolic energy and they are also readily soluble in water. So you can see electron micrograph image of adipose tissue. Each fat globule occupies nearly the entire cell. You can see here. hmm, This slide you may also be asked in anatomy to identify the tissue. The signet ring appearance. Fat globule has occupied almost the entire volume of the cell, pushing the nucleus to one side, giving it a signet ring appearance. While making the slide, the fat dissolves and leaves an empty vacuole, right? And this is how typically white adipose tissue looks, right? What is brown fat? Brown fat or brown adipose tissue is where it is like rich in mitochondria, and the mitochondria have large amounts of iron that gives it brown or beige appearance. And what is the important feature of that is the oxphos that happens there, like oxidative phosphorylation happens in mitochondria, but in brown adipose tissue, oxidation is uncoupled from phosphorylation because of uncoupling protein, thermogenin being one of them. And so that uncoupling protein, NADH and FADH2, they give their electrons to the ETC. Electrons flow through ETC and protons are pumped from the mitochondrial matrix to intermembrane space, creating a proton gradient. But the protons do not flow back into the mitochondrial matrix uh, via the ATP synthase. They rather flow through separate channel, and that is uncoupling of oxidation from phosphorylation. And so when they flow back into the mitochondrial matrix through a separate channel, which is created by the uncoupling protein, the energy is still released, right? Water, when it falls from a great height, there will still be energy which will be released. So this energy which is released when protons flow along their concentration gradient, it gets dissipated as heat rather than forming ATP. And generation of heat is the main purpose of brown fat to prevent hypothermia from happening. Like especially these animals, right, who are in cold climates, they have a lot of brown adipose tissue. Even human beings, especially the babies, have a lot of brown adipose tissue compared to adults because for that only, brown adipose tissue is necessary for adaptive non-shivering thermogenesis. There is shivering thermogenesis due to muscle contraction, and non-shivering thermogenesis is in brown adipose tissue. That declines with age because of all of these factors which are associated with aging, like mitochondrial dysfunction, reduced sympathetic nerve signaling, decreased function of adipose or adipose stem cells or progenital cells. All of these are seen normally in aging, and these are responsible. Additionally, progressive hormonal changes and chronic low-grade inflammation, right? Progressive hormonal changes and lowering of sex hormone levels as you age. And there is also chronic low-grade inflammation associated with aging that cause remaining brown adipose tissue cells to shift towards a white phenotype, reducing energy expenditure. Now, at this point, I would like to point out that the number of adipose tissue cells in an adult human body remains approximately the same. There is a dynamic uh, turnover that happens in the sense that about 10% of the cells of adipose tissue die every year, and every year they are replaced Similar number of cells are replaced. So overall number of cells in a human body, adipose tissue cells, remains the same, right? So what happens when we gain weight or lose weight is that the cells, when we gain weight, they swell up with fat, triglycerol. When we lose weight, the cells shrink in size. That is what happens when we gain or lose weight, or when we gain fat or lose fat. But number of adipose tissue cells remains constant in an adult, more or less. And this may be the principle behind surgeries like liposuction, right? And brown adipose tissue is still located. So this what can happen is, like, this brown adipose tissue can become white adipose tissue. White adipose tissue can get converted to brown adipose tissue. That is what all can happen, but number of cells of adipose tissue overall remain the same. Now with age, because of all the age related stuff, brown adipose tissue goes down, but still some of it is located in supraclavicular area, like about 100 to 300 gram is there in the body, right? And and it's found in supraclavicular area, neck, upper chest, kidney, around the kidney, adrenal, and spinal column. What are the advantages actually? We saw that white adipose tissue will store triglycerol, right? And actually, then that is all associated with all the bad effects. Whereas brown adipose tissue burns fat, or it burns fat basically without uh, leading to ATP formation, and that fat which is burned, that energy is released as heat, so that we are actually having fat loss. Because fat burnt with ATP, the ATP can again be used to synthesize fat and other stuff, but here fat is lost as energy, as heat, right? So brown adipose tissue actively burns energy to generate heat, and unlike white adipose tissue which simply stores it, brown adipose tissue helps in calorie burning. Up to 300 calories a day can be burned by brown adipose tissue to maintain body temperature. Metabolic health: brown adipose tissue breaks down blood sugar, regulates blood lipids, reducing the risk of obesity, type 2 diabetes. It cleanses the bloodstream of excess amino acids like leucine, lowering metabolic disease risk. How do we increase brown fat percentage in our body? I mean, brown fat can increase in our body by conversion of white fat into brown fat. White fat can be stimulated to become brown like, right? That can happen by these mechanisms like cold exposure, exercise, muscle movement, certain foods, and adequate sleep, right? Thyroid hormones being adequate, and compounds like GLP that act on GLP-1 receptors, semaglutide, right? So these can help to increase the brown fat in the body, and these measures or these things can also activate brown fat which had become inactive. So increase in brown fat is by two pronged. One is activation of inactive brown fat, and transdifferentiation of white fat into brown fat. Now coming to this partial hydrogenation of oils. Oils are unsaturated, right? Now if you hydrogenate them, their shelf life increases because they are then solid at room temperature, right? Now there are natural fats at 25 degrees. Olive oil has a lot of unsaturated fatty acids, C16 to C18 unsaturated fatty acids. Butter has slightly more saturated fatty acids. Beef fat has even more saturated fatty acids, right? And higher portion saturated fatty acid in butter increases its melting point, so it is solid at room temperature. The point is that partial hydrogenation of commercial vegetable oils was used to improve shelf life and increase the stability of cooking oils at high temperatures used in deep frying. This process converts many of the cis double bonds in the fatty acids to single bonds and increases the melting temperature of oils so that they are nearly solid at room temperature. Partial hydrogenation, however, has an undesirable effect in that some cis double bonds can be converted to trans, and there is now strong evidence that dietary intake of trans fatty acids, often referred to as trans fats, leads to higher incidence of cardiovascular disease. How? Because trans fats raise LDL and they lower HDL, and they also increase inflammation in the body, right? And that is why regulatory agencies in the world now are banning trans fats in packaged foods. And that dalda, that is one hydrogenated vegetable oil. It's not good for our health. When lipid-rich foods now, that was trans fats. Coming to rancidity. Rancidity comes from some Greek word which actually means rotten or to get spoiled or something. So rancidity is fats which, especially unsaturated fats, which get ruined or spoiled by exposure to air or oxygen, and then they give a bad smell or foul odor. That foul odor is one of the meanings of this origin of this word of rancidity as well. So when lipid rich foods are exposed to too long to oxygen in the air, they may spoil and become rancid. Unpleasant taste smell associated with rancidity results from oxidative cleavage of double bonds in unsaturated fatty acids, which produces aldehydes, carboxylic acids, and shorter chain length and therefore higher volatility. These pass readily through the air to the nose. Now, this is the mechanism of rancidity. There's initiation, elongation, termination, like maybe like transcription, translation की तरह इसका हो रहा है, initiation, prolongation, termination, right? Ultimately, it leads to these compounds which are toxic and even they give this bad odor, right? This is also another mechanism, like mechanism in another way of expressing. This is a like beautiful summary of rancidity. Spoilage of food containing fats and oils due to oxidation, causing unpleasant smell and taste, right? Oxidation of fats and oils when exposed to air. Fat plus O2 oxidized products, bad smell. How do you prevent it? By storing it in airtight containers, refrigeration, adding antioxidants, nitrogen gas flushing in chip packets. Nitrogen is an inert gas. It will prevent oxygen from spoiling the fat in the food. This prevention of rancidity is prevention of food wastage and prevention of health hazards because of the toxic chemicals which would be there because of the fat becoming rancid or spoiled, right? Cis and trans fats. So you see cis fats have this kink or bend because of which the melting point is decreased and they are liquid at room temperature because they are not able to be tightly packed. Whereas trans fats, where the groups are on opposite side of the double bond, they are more like saturated fats. Then and that's why they become like solid at room temperature and they're not good for health. This is the difference between rancidity and hydrogenation, which is done commercially. You can pause and read. Rancidity and hydrogenation are both oxidation, but hydrogenation is addition of hydrogen to unsaturated bonds, whereas rancidity is a natural process which happens by exposure to oxygen. It is oxygenation, right? And hydrolysis, whereas rancidity is addition of hydrogen, right? Actually, they are opposite commercial process of handling fats. Rancidity is spontaneous spoilage of fat, whereas hydrogenation is a controlled industrial process that alters fat chemically to improve its texture, shelf life, resistance to that very spoilage. This you can read this as well. Coming to the last part, waxes. Waxes are esters of long chain fatty acids, long to very long chain fatty acids, right? And saturated fatty acids or unsaturated, whatever, with long chain alcohol. So ester of long chain saturated or unsaturated fatty acid with long chain alcohol are waxes. and they have melting point of about 60 to 100 degrees Celsius, right? They have a higher melting point, so they're definitely solid at room temperature, and they have a diverse function like water repellent, right? We all use wax as a moisturizer, like that Vaseline we use as moisturizer, Vaseline lip balm, and that's a natural waterproof compound of nature is wax. Birds birds have their feathers covered in wax, which acts as a water repellent. Some leaves like lotus leaves, etc., they have wax, right, which prevents excessive evaporation of water. And also wax protects against parasitic infection. How does it protect against parasitic infection? Firstly, it's a physical barrier, and it is a water repellent, prevents pooling of moisture which is required for growth of fungus. And the wax can also contain antimicrobial chemicals which directly kill the microbes. So biological wax, now this is palmitic acid with a long chain alcohol. This is like 30 carbon alcohol. Tricontanol. Biological waxes find a variety of applications in pharmaceutical, cosmetic, other industries, blah blah. They are used in lotion, ointment, right? Hmm. Lipids. Okay, to summarize, lipids are water insoluble cellular components of diverse structure that can be extracted from tissues by nonpolar solvents. Almost all fatty acids like have hydrocarbon components. You can just read the summary actually, and most of the fatty acids have even number of carbons, usually 12 to 24, and double bonds are almost always in cis configuration. Triacylglycerol molecules are esterified to the 3-hydroxyl group of are triacylglycerol are fatty acids esterified to 3-hydroxyl groups of glycerol. Simple will have one type of fatty acid, mixed will have two types of fatty acid. Then triacylglycerols are used in storing energy and providing insulation. Um, and then fats can turn rancid, or partial hydrogenation has been used to extend their shelf life. This process produces trans fatty acids, partial hydrogenation, which is increasing the risk for coronary artery disease, and their uses in prepared and processed food has become highly regulated. Waxes are esters of long chain fatty acid with long chain alcohol, with diverse uses in biology and industry. With this, we come to the end of this lecture. Next, we'll see the other classes of major classes of lipids. And I'm sorry I'm recording this lecture quite late at night and I'm kind of sleepy. But I tried to cover the concepts wherever there were concepts, and where it was just you could read the slide, you could read the slide. If there was anything conceptual, I've tried to describe it separately. If you still have any questions, please feel free to comment and I will answer the question to the best of my ability. Thank you. Have a nice day.